For example, that what we do is we collect all the target-based assay result and cellular-based assay result. And then we build the classification model for all of them. And then we only choose those data that it generates a model has a AUC larger than 0.85 that we think is a good classifier that we precede the modeling process. So that so basically we build two global antiviral model based on mixed type of data against the different virus species, and then the second class is they're different species, but they have a target in common that we build the target-based models that even their species are different, and we test the performance and do standard five-fold cross-validation, use that to screening new possibilities. So which here that once the model is built, we reach to the last step, that screening new possibilities. So here, normally there's, for virtual screening, there are two paths right now we can do. One is a data-driven AI model that we just discussed. Another is once we have the target structure available, we can use conventional computer-aided drug design, CADD technology, to do docking and rational design as well. So that in-house we normally push the two routes together. So for the short term, we can screening we can perform basically drug repositioning practice, screening FDA approved or drugs that is in clinical trial readily to give to patients, or even nutraceuticals or natural product that can be purchased directly. And for the long-term project, we normally would screen a commercial compound library. Right now, the global commercial supplier can supply up to billions of level of new chemicals ready to purchase. And for the virtual library, it's hundreds of billions level of molecules. So that, but for computation to screening that level, it doesn't really costs as much time as in the past, since the computing technology is so advanced nowadays. But we just talked about small molecule earlier, but how about antibody and vaccine? So that for viral infections, we would assume that antibody is in general a better option because it's more specific and more effective. And also vaccine is really the long-term solutions to prevent the current or the future outbreak. So those two type of effort is very important as well. And to assess a successful antibody or vaccine, first we have, of course, we have to assess the specificity of those designed biologics again that mimic the immunology process of the new virus, and also its immunogenesis for vaccine and antibody are different. So we have to assess them in different standard, and toxicity are important for both. So as we can see that earlier, this picture shows this is the S protein and this is the human ACE2 binding. And we also know that the antibody, the neutralization antibody also binds to this, we call the receptor binding domain primarily for inhibit its activity and neutralize the virus basically. But however, the virus are constantly evolving species, and most of the mutation residues normally located on the surface of the protein, because that's where it interacts with its environment and triggers a more diverse profile. So in that case, we really need a real-time mutation track. And then based on the mutation data, we can further using computational immunology technique. So basically it's a combination of bioinformatics and immunology and also structural biology technique to to rationally design vaccine and antibody. So it's also a technology that some part AI can play a role, such as predict its neutralization effect, immunogenesis, or toxicity. But here I would like to introduce a very important piece of the technology where in the AI domain as well is this real-time tracking for the mutation through time and location. So here is also, this is another GitHub project called the NextString. So NextString is initiated by the Swiss Institute of Bioinformatics and later being participated by dozens of different scientists all over the world. That what they do is they pull the gene sequence information from GISAID, which is a temporary repository for infectious disease sequence. And then they pull that on a frequently, like hourly or daily basis. And currently, as of today, April, there is over 4,000 genomes sampled. And you can see how they are different over time, and they're different over location. And over here, this graph shows the diversity of the basically where the location happened, and in each different positions on the genome. And you would and look, this is the S protein, which is where we our vaccine and antibody effort primarily focus on. Also, very already being mutated very in a large degree, so that this is really a good project for us to real-time track if those regions were originally designed based on have already mutated into new residues that may compromise our initial discovery effort, and that's a very important piece of information. So, and this is also a project that's nearly a digital twin. So a digital twin is a concept probably new to people in biology field, but it's frequently used in like aviation and other industry field. Basically, digital twins means a digital replicate of a physical identity and a physical entity. It can be a living being or a non-living entity. So it could be a digital twin of an airplane, a car, but it can be a digital twin of a person or a virus in this case. And it has to evolve over time, that mirroring every state of time and this its primary physical entity is. So that ideally, if we can leverage all the technologies I have just mentioned earlier to create a digital twin of a novel virus, we will be really in a good ground on tracking its behavior and design better medicines for it. So here's just a quick recap of the entire workflow. So first we query the gene sequence, and then we mine all the data that is relevant. And then based on those data, we reasoning and do further inference. And then we come up with the top list, and then we go on modeling and finding new possibilities of therapeutics. So this entire workflow are most of the programs are already pretty mature, and a lot of them are done using Python scripting. And it can be, it's not completely integrated yet, but I think it will be very helpful that if we integrate all process together and make it more efficient. So here is some conclusion here. So AI has many potentials in finding treatment for unknown and new pathogens because it's fast. We know that computer takes much faster to compute than human, right? And also it's broad because it scans all available information and in a much shorter time frame than human. So human may missing evidence easily or just cover very small portion of the entire evidence scope, but AI can cover basically every corner of the information. And also it's rational. It judges every evidence equally, unlike human beings, they may judge the things they're more familiar with a higher weight. And also AI is data dependent. So this is very important, so that without data, AI won't perform very good. So that in a global pandemic like this, requires global collaboration on data sharing and fast community mobilization for efficient research and iteration. Then there is still limitations in AI technologies in drug discovery that requires considerable amount of human intervention. So basically each steps at the time that it's supposed to be AI is reasoning, but sometimes we reason for it because the rule hasn't been completely written in it. So that it needs a very deep understanding of the disease biology and the drug discovery logic to design an expert system to help the AI to do the reasoning work. And also it needs considerable engineering effort to integrate all the process flawlessly. But we believe it can be achieved in the near future. So ideally, if we were able to create a digital twin of any new pathogens based on initial limited but constantly evolving data, we will be able to design better therapeutics in time to control the pandemic. So that's my talk. Thank you, everybody.